Hello, my name is Chang from the Uiversity. In this session, we're going to look into the science of the SARS-CoV-2. In this session, we are going to talk about the active prevention and the therapeutic mechanisms and the development of the vaccine. Also, we're going to look into the science behind the COVID-19. By thinking about that, we're going to look into what can be the best therapeutic methodology or the approaches we can take. In this session, we are going to invite the experts who studied a lot about the virus and the infectious diseases. So we have the three speakers. We have the Professor Shin-Ru Shih from the Taiwan's Chang University, and the second speaker is Professor Chang Shih from the Seoul National University, and the third speaker is from the Northwestern University. We have Professor Judd Hartquist. We have three speakers.
Let me begin the first session or first seminar, and the first presenter or speaker is from Taiwan. She is from the Chang University, and her name is Shin-Ru Shih. Let me introduce her briefly. As for Professor Shin, she was from National Thai University, and after graduating the school, and at the same graduate school, she studied biochemistry and went over to Berkeley University in the USA, got a PhD degree, and later Center for Technology and Medicine in New Jersey is where she got the postdoc, or finished the postdoc course. From 2004 and onwards up to the present, she is with the Chang University at the Department of Lab Science. Viral infection, infectious diseases are the main topics of her paper, which amounts to 170 or more. This means that she has been engaged in the studies for many years. And today she is going to talk about the virus and host, clinical and basic research for COVID-19. And she is going to cover the researches done in Taiwan and also share the scientific information with us today. Without further ado, I'm going to give the floor to her.
Hi, everyone. Thank you for listening my talk. And also thank the organization committee from VIXI invite me as an online speaker. My topic is virus and host, clinical and basic researches for COVID-19. My talk here will focus on two parts. First is laboratory diagnosis, and the second is development of antivirus. For laboratory diagnosis, I will present two work, and the first one is culture-based virus isolation to evaluate potential infectivity of clinical specimens tested for COVID-19, and we published in Journal of Clinical Microbiology recently. This work, I'd like to thank the research team from Clinical Biology Laboratory, Chang Memorial Hospital. We are the CDC contract laboratory, and we are in charge in laboratory diagnosis for COVID-19, especially for the imported cases from airport. And this study were majorly done by Dr. Guoming Li and Xionghui Huang. We collect 60 specimens from 50 patients from January 25th to March 21st. The specimens including the swab, nasal swab, and the sputum. And some of the specimens were kept in -80, so they underwent frozen and thaw procedures. And in comparison with the freshly collected specimen, the culture time in general is from 3 to 9 days. And the frozen and thawed specimens got more longer time, from 9 to 18 days. We use real-time Rtbcr for laboratory diagnosis. The protocol is based on Taiwan CDC, which is similar to WHO guidelines. We detect viral NSP12 gene, which is viral RDRP polymerase gene, and also envelope gene, E gene, and the nuclear capsid gene, N gene, for each sample. And in this figure, Figure A shows viral RNA copies numbers in the specimens, which can be cultured, and Figure B shows RNA copy numbers in the specimens, which cannot be cultured. The scales of the vertical axes are the same. In comparison of Figure A and B, it's clear in general if RNA copy is high, virus can be isolated from the specimens, as expected. On our hands, specimens containing at least 10 to 5 power RNA copies, which can be cultured from the specimen. The data is consistent with several other studies from different countries. Besides RNA copy number that would affect culture rate, we found genome integrity is another important determinant. In figure C and D, the vertical axis is the RNA copy number of NSP12, a genomic RNA. The horizontal axis is the RNA copy number of E or N. It's subgenomic RNA. Culturable specimens have better linear regression than that of non-cultural specimens. We compare the red line with that of blue line. And in figure E, we particularly compare those frozen thawed specimens. Frozen thaw has been considered a procedure that may break viral RNA. And indeed, we found that culturable specimens have a good linear regression between genomic RNA and subgenomic RNA. Very poor linear regression in non-culturable specimens, indicating that genome integrity is a key determinant to know whether live virus existed in a specimen or not. So for this study, we got the conclusion as following. The lowest copy number required for virus isolation was determined to be around 5.46 and 5.7 log genomic copies per ml sample for detecting of NS12, E, and N gene, respectively. Culturable specimens are characterized by high copy number with a linear correlation observed between copy numbers of applicants targeting structural and non-structural genomes. It's genomic RNA and subgenomic RNA. In addition to the copy number, the integrity of the viral genome should be considered when evaluating the infectivity of clinical SARS-CoV-2 specimen. Now I move to the second part of laboratory diagnosis is we do the genomic surveillance in Taiwan, and we found a novel ORF deletion mutant and a nucleate possibly associated with infection in Middle East. And this paper has been published recently in Emerging Microbes and Infections, usually done by Dr. Gung Chen and Dr. Gung. We further performed sequence analysis of SARS-CoV-2 isolates in Taiwan. We found an interesting variant in which ORF8 was deleted in one isolate from a business name, came back to Taiwan from mainland China in the end of January. Figure A and B are the sequences analysis by NGS, and we found the peak around 3' end of viral genome was different from the others. We further used Sanger sequence to prove there is an ORF8 deletion in the isolates. The similar finding was also reported by Singapore group. And this figure shows phylogenetic tree of Taiwanese and global strains. Basically, most virus isolates were from imported cases. Therefore, as expected, the sequences are similar to the region they came back from. For example, China, other Asian countries, Europe, or America. We use this simplified phylogenetic tree to explain sequences of Taiwanese isolates in public. As mentioned, the imported cases cluster with the isolates from the regions they came from. Here we found our several local cases in this yellow cluster. They were clustered together with some isolates from travelers who have travel history of Middle East countries. Not too many full genome sequences of virus isolates from Middle East countries. Therefore, we believe we kind of contribute to more sequences in this nucleate. So in this work, we found 882 nucleotide deletion in ORF8. And this was found in one isolate in the first wave. And this deletion does not affect viral replication in cultured human cells, which I have not shown the data here. And we got 5 clustered CGMH or hospital cases. And we believe it belongs to a nucleate harboring ORF1AB B378I mutation, and a group with some strains from patients have the travel history to Iran and Turkey. We believe that genomic surveillance contributes to a better understanding of the global SARS-CoV-2 transmission dynamics. Now I'd like to present a work, is that antibody drug, and we published this paper in Nature Structural and Molecular Biology recently. It's about the structural basis for the neutralizing of SARS-CoV-2 by an antibody from a convalescent patient. This work majorly was done by Dr. Guanying Huang in collaboration with researchers in Oxford. We isolate virus specific B cells from COVID-19 patients and then did immunoglobulin cloning and then transfection, expression, and purification. We obtained many human monoclonal antibodies from COVID-19 patients. Here I'd like to present an antibody called EY6A. Here we focus on one monoclonal antibody called EY6A. We analyzed the binding affinity for different viral surface protein. The black line is SARS-CoV-2 spike protein. The red line is SARS-CoV-2 S1 protein. The blue one is SARS-CoV-1 S1 protein. The blue one is MERS spike protein, and the orange one is OC33 spike protein. So for the antibody isolated from COVID-19 patient, got highest binding affinity for SARS-CoV-2 spike protein, and also very high affinity for S1 protein. Moderate binding affinity for SARS-CoV-1 S1 protein, and no binding activity for MERS and OC33. Back to the figure B, and we also use another antibody called CR3022 as a control. This antibody was isolated from SARS patient 17 years ago. So it got the highest binding affinity for SARS-CoV-1 S1 protein, and moderate binding affinity for SARS-CoV-2 spike and S1 protein, and also no binding activity for MERS and OC33. And this is from patients. And we found the patient serum got a very high binding affinity for SARS-CoV-2, as expected, and also moderate binding affinity for SARS-CoV-1, and low affinity for MERS and OC33. Figure D is immunofluorescent assay, and we can see EY6A got a signal indicating that EY6A can bind to SARS-CoV-2, a surface protein. And we also use influenza monoclonal antibody as a control, and no signal here. So these results indicate the specific binding of EY6A by ELISA assay as well as immunofluorescent assay to SARS-CoV-2. And we then test antiviral effects for this antibody. And also we use the CR3022 as a control. And we can see when we add EY6A to the culture medium, and we analyze the viral RNA copy numbers, and it inhibits a lot because it shifts to a higher CT value, indicating the RNA copies was lower. And we also use human IgG as a control, and we cannot see any effect. And we also use a plaque reduction assay, and we found indeed this EY6A antibody can inhibit plaque formation. And this is a comparison with CR3022. And we can see EY6A have a better inhibitory effect for SARS-CoV-2 than CR3022. And this work was majorly done by Dr. Peng Huang at P3 Laboratory. And from EM structure, and this was done by Oxford University research team. And we got the structural base to know the interaction of EY6A. And majorly, it interacts with receptor binding domain, but not exactly the binding domain. Also covers some regions that outside of the receptor binding domain, indicating that probably receptor binding domain alone is not a determinant for a monoclonal antibody that can neutralize the virus. Now I'd like to switch to the last part is about the small molecule drugs. The first one is we identified 2 FDA-approved drugs inhibit human enteroviruses and coronaviruses. And we also discovered an M-protease inhibitor encoded by SARS-CoV-2 that can inhibit SARS-CoV-2 replication. We have been using ADCL virology platform to screen key host vectors involved in RNA-virus infection. The platform is based on CRISPR-Cas9 technology. CRISPR vector containing sgRNA was transfected for constructing sgRNA library lentivirus. After transduction to cells, we obtain sgRNA library expression cells, and then we challenge these cells with RNA viruses. RNA virus infection causes cell death. If cell survived after virus challenge, indicates the cell lost key vector for virus replication. By such platform, we identify several host vectors that are important for enteroviruses and coronavirus infections. Here we focus on this factor A, which involved in lipid metabolism. We therefore tested several inhibitors targeting to this metabolic pathway to know whether virus replication was reduced. Indeed, in comparison to control, indicated by black line here, we found these inhibitors can significantly reduce viral titer, either enterovirus or coronavirus, 29E and also OC33, which can cause common cold in humans. And very importantly, we tested the 2 of the 4 inhibitors. Why we choose these 2 inhibitors? Because it is FDA-approved drugs, which has been used in other diseases. And these 2, we found it can dramatically inhibit viral replication. When we use real-time Rtbcr, either ENG or RDRP gene, we can see the reduction at least 1 log. And we also perform plaque reduction assay. And in comparison with the control, we can see many plaques here. When we add these 2 inhibitors, we can see the very great inhibitory effect to reduce SARS-CoV-2 replication. Finally, I'd like to introduce an inhibitor which inhibits SARS-CoV-2 protease. And here was done by Dr. Shin-Ru Shih's team and also my team members, Dr. Huang, Shen-Yu Peng, and also Dr. Gung. We use cDNA synthesized for the M protease, and we use GC376. Actually, it has been done for feline coronavirus. It's a great inhibitor for that virus. And we got the M protease, and we established the assay, and got this one is a very good potent inhibitor for SARS-CoV-2 and protease. This is the cleavage assay, and we found it can inhibit the M protease, also called 3C-like protease. And this is the Ki value is about 12 nanomolar, quite potent. And the IC50 is about 26 nanomolar. And we then test this inhibitor, GC376, on SARS-CoV-2 virus replication. And this is when we measure the TCID50, we can see the very great inhibitory effect for this inhibitor. The IC50 here is around 0.91 micromolar, and CC50 larger than 100 micromolar. And this has been published in Antimicrobial Agents and Chemotherapy recently. And we believe it can be further developed as an antiviral for SARS-CoV-2. So for the small molecule drugs against SARS-CoV-2 replication here, we identified several small molecules. The first two is FDA-approved drugs. It can inhibit viral replication, targeting to host gene. And another one is targeted to viral protease, and we hope it really can be developed in the future as antiviral agents. So I'd like to give a summary of this talk. For laboratory diagnosis part, we used a culture-based virus isolation, and we evaluated the potential infectivity of clinical specimens tested for COVID-19. And we found not only the viral RNA copies, but also viral RNA integrity is important to determine whether virus existed in the specimens or not. And we also did the genomic surveillance of SARS-CoV-2 in Taiwan, and we found a mutant virus which got the ORF deletion mutation. And we also found several local cases clustered with some isolates from the visitors who have travel history from Middle East country. So this nucleate possibly associated with infection in Middle East. And for development of antivirus, we got an antiviral drugs, and which have a very great neutralization for SARS-CoV-2. And we also identified several small molecules and two FDA-approved drugs which can inhibit human enteroviruses and coronaviruses. And we also discovered an protease inhibitor encoded by SARS-CoV-2, and which has been used in animal coronavirus, and we hope it really can be further developed as an antivirus in the near future. Finally, I'd like to thank those people, the Chang COVID-19 research team from Chang University and Chang Memorial Hospital, and also the team members in National Health Research Institute, and also we have collaborators from Taiwan Academic Clinical, and also team members from Oxford University and also Rutgers University in the United States.
Thank you very much for the wonderful lecture. And in the early 2020s, there was a really excellent research that was going on within the Taiwan and about the treatment and the vaccine. The virus isolation is really necessary. She talked about the virus isolation and some of the mutations that was found on the field. And also she told us about the platform that can screen the virus replication. And recently, we are talking about the therapeutic candidates that can contribute to the early termination of the COVID-19.
Among those, along with the other pharmaceutical companies, I believe that you are conducting clinical trials. As far as I know, I hope that we have a promising outcome sooner than later. Thank you so much for your wonderful presentation, Dr. Shin-Ru.